All right, I'm going to start chapter 10. If you guys took your books home, great. I didn't, so I'm like, oof. Anyway, this chapter is going to be a lot. So the first thing I'm going to do today is talk about how DNA was discovered. Because a lot of times we take for granted the fact that like DNA, like everyone just knows, well, most people know what it is. I have seen one video where one woman, she's an anti-vaxxer, says that the COVID-19 vaccine, quote, snaps DNA in half.
Oh,
Yeah. And
what?
I'm just like, oh my God. Like, and she wasn't even American. She was German, living in America. But I guess
How do
maybe
you
just,
snap DNA in
I
half?
don't know. Like I said, I'm just like, people are, oh, so dumb. Anyway, so discovery, like how DNA was discovered, how we identified the molecule, and also how we figured out that it is responsible for being the genetic information. So like we already talked about heredity and how genes are passed down, characteristics are passed down from parents to offspring through Mendel's genetics. Now we're going to look at how the code in DNA relates to like a physical meaning, as in like making proteins, because all of your traits are due to the actions of proteins, and it acts all through an intermediate called RNA. I gotta find my pencil. Where did I put it? So that's why it's called DNA, RNA, and protein synthesis. So basically, the way that it works in general, the instructions to make protein are on DNA, but DNA is in the nucleus, right? So if I have a cell here, here's the nucleus. DNA is in the nucleus, but proteins, synthesis. Proteins are made in the cytoplasm, and the DNA cannot leave the nucleus. So there has to be something that can basically copy the code for the protein from DNA, leave the nucleus, and go to the cytoplasm to make protein. And that is a molecule called RNA. So that's the broad stroke, big picture. Obviously, it's going to get a lot more complicated than that. This chapter and the next chapter are some of the most like intricate, like most detail oriented type instructions, information. But we're just going to take it a step at a time. I don't even think we're going to get through the whole chapter, hopefully by the end of next week. And then after that, I'll see you back at school in person. But for today, it is just on the discovery of DNA and the experiments that laid the foundation for determining that DNA is responsible for all of our characteristics. So the last thing we talked about was Mendel. And from his studies, he concluded that hereditary factors determine an organism's traits, right? Those are the alleles. What were these hereditary factors? How did these molecules store hereditary information? Mendel was only there to really kind of say, okay, I know that there's these things that control traits, and there's different versions of them. There's dominant versions and recessive versions. But he didn't know more specifically than that, more specifically than like these factors, like he said, these hereditary factors, alleles. Scientists believed that if they could answer these questions, they could understand how cells pass on characteristics to their descendants. Their answers to these questions began to emerge during an epidemic, speaking of epidemics, of pneumonia in London in the 1920s. Okay, so the first experiment that we're going to look at is Griffith's experiment done in 1928 by Frederick Griffith. There he is, of course, old white dude. He was studying a bacterium called Diplococcus Pneumoniae. Diplo obviously meaning two, coccus, cocci is a size, and bacteria meaning round. There's three different shapes of bacteria. There's coccus bacillus, which is rod-shaped. Bacillus, and there's Spirulina, which is spiral like that. This one is very rare, Spirulinea, but we can basically classify diseases that are caused by bacteria partially based on their shape. Anyway, Griffith was trying to develop a vaccine against pneumonia. Now, there's actually different kinds of pneumonia. There's bacterial pneumonia and viral pneumonia. And obviously we can't, what do you call it? We can't treat viruses with antibiotics because if you look at the word antibiotic and break down the word into its parts, we have anti, bio, and tic. So this is an adjective. Bio means life and anti-life, obviously. Viruses aren't really alive, so antibiotics don't work against viruses. That's why we use vaccines instead. Okay, so he studied two groups of bacteria. Now, the virulent bacteria, or the bacteria that cause pneumonia, is surrounded by a capsule that's made of polysaccharides. Now, that capsule protects the bacteria against our immune system. And under the microscope, these colonies appear smooth because of the capsule. So he called them the S strain, S for smooth. Okay, so these are the disease-causing bacteria. The S strain for smooth. Now you can see on the other side, you have the rough colony, or the R strain. They call it benign, meaning it's not disease-causing. And you can see from the caption, it lacks a protective capsule. It is able to be recognized by our immune system, and therefore our immune system can destroy it. The capsule, you can think of it like an invisibility cloak or like a shield around the bacteria. For the S strain, our immune system can't get through it. So the second strain of Diplococcus does not cause pneumonia. It does not have a capsule, and so it appears rough under the microscope. We call that the R strain. Okay, so S strain, bad. R strain, not bad. I'm not going to call it good because it's still bacteria. All right, so here's what Griffith did in his experiment. He used these two strains, the S strain. So I'm just going to put it again up here. S strain leads to pneumonia. R strain, no pneumonia. And he did 4 different experiments, which I have down here. So for experiment number 1, he injected the mouse with live R cells. Remember, R is no pneumonia. So he injected the mouse with live bacteria that was rough, and the mouse lived. Experiment number 2, he injected a mouse with live S cells. S creates or causes pneumonia. And of course, the mouse developed pneumonia and died. Experiment number 3, he took the S cells here and he did something called heat kill. And what heat kill means, a lot of our vaccines are actually heat killed. So what that means is when you expose DNA to high temperatures, like around 50 degrees Celsius, DNA does something called denature. Oh, I cannot spell. Denature. And it basically means to fall apart, falls apart. Now, you know the characteristic shape of DNA is the double helix. And if the DNA doesn't have that shape, then it's difficult to read. Basically, just kind of, like I said, it falls apart. If any of you guys like have a knitted sweater or something like that, and you pull on a string and you just keep pulling and pulling and pulling and the whole sweater falls apart, that's kind of like denature. So the only way that bacteria and viruses can cause illnesses if, number 1, they can get into your body, number 2, their genetic material starts to alter what happens inside your body. So if you destroy the DNA of the bacteria, then the what do you call it? The theory is that it won't cause any problems in your body. So for experiment number 3, he killed the S cells with heat and injected the mouse with those heat-killed S cells, and the mouse survived. So that makes sense because we're basically going to expose the bacteria to high temperatures, which basically kills the bacteria. Then we're going to inject the, let's call it not quite dead or heat-killed bacteria into the mouse survives. Here's the weird part, okay? Experiment number 4. He killed the S cells with heat, but he mixed them with living R cells, okay? So we have heat-killed S cells and living R cells. Now, under individual conditions, through experiment number 1 and experiment number 3, live R cells result in a mouse surviving. Heat-killed S result in the mouse surviving. But when you mix heat-killed S with living R, the mouse died. So like, what? That makes no sense, right? If you take this result and this result, they both survived, but you mix the two experimental groups together, living R cells, heat-killed S cells, and you ended up with a dead bunch of mice. So he discovered, oops, let me take that away. He discovered something called transformation. And now transformation only occurs in bacteria, but it is the ability of bacteria to alter their genetics by absorbing foreign DNA from another bacteria. You just think of it that basically the heat-killed S cell basically turned the living R cell into an S cell. Okay, and you can see that from, I've got a better shot of this. So you got S cells killed with heat, which up here resulted in nothing. You have living R cells, which in experiment 1 resulted in nothing. But you mix these two together, you inject the mouse with a mixture, they died. So he concluded that hereditary material from the S cells transformed the R cells into a virulent strain or a pneumonia-causing strain. And the R cells, even though they were harmless up here, they became deadly when mixed with the heat-killed S cells. That process is called transformation.
Have you ever wondered what determines your height, hair, and skin color, and even the size of your nose? It's DNA, the genetic material of cell. But do you know how it has been proved? Here is the experiments that proved it. Griffith's experiment. Griffith took 2 strains of pneumococcus bacteria, S3, which have smooth polysaccharide capsule, giving smooth colonies S, and it's virulent, causing pneumonia, and R2, which have no capsule, giving rough colonies R and is avirulent, unable to cause pneumonia. When the virulent S3 bacteria injected in mice, it killed them, causing pneumonia. But when the avirulent R2 injected in mice, the mice lives. If you injected the heat-killed virulent S3 bacteria, the mice still live, implying that live S3 bacteria are needed for causing pneumonia. But interestingly, when mice were injected with the mixture of heat-killed virulent S3 bacteria and avirulent R2 bacteria, mice died. Analysis of dead mice reveals live S3 virulent bacteria, suggesting something from the heat-killed S3 had transformed to the live avirulent R2 bacteria, responsible for the transformation of the live avirulent bacteria to virulent S3 bacteria, causing death of mice. This is called as bacterial transformation. To prove DNA...
Whoa. Oh, okay, I guess that was it. Right, so like he said, basically something, and this is still the question now, something, that's the big question mark, some kind of hereditary material went from the dead but virulent S bacteria and transformed the R bacteria into an S bacteria because it added the capsule. The question really now is, what is that hereditary material? So now we have the next experiment, which is done by Oswald Avery. There you go, another old white guy. This is in the 1940s, so it was like more than 10 years later. He tested whether the transforming agent was protein, RNA, or DNA. He used enzymes to separately destroy each of those three things. So we had an enzyme that destroyed protein, an enzyme that destroyed RNA, and an enzyme that destroyed DNA. And then he mixed them with the heat-killed S cells, and he separately mixed 3 experimental batches. He found that cells that were mixed with protein and RNA were able to transform the R cells into S cells, but cells that were missing, sorry, I did not describe that properly. Let me try again. So we've got 3 groups. All 3 groups have protein. All 3 groups have RNA. And all 3 groups have DNA. The only difference is in let's say the first batch, he would mix it with an enzyme that would digest protein. And the second batch, he mixed it with an enzyme that destroyed RNA. Now for these 2 batches, the R cells were still transformed into S cells. But for this one, the last one, he had an enzyme that destroyed DNA. And for that one, the cells that were missing DNA did not transform. So the R cells stayed R cells. They did not cause pneumonia. So this experiment by itself should have convinced the scientific community that DNA was in fact that genetic or the hereditary material that was transforming. But like a lot of things in science, sometimes you need more than one experiment. So it wasn't just Avery's experiment that convinced everybody. The last experiment that really kind of solidified the idea that DNA is a genetic material for the scientific community was about another 10 years later. Finally have a woman, woohoo, Martha Chase and Alfred Hershey tested whether DNA or protein was the hereditary material that viruses transfer, because it turns out viruses can do transformation as well, but they don't do it between viruses like bacteria do. They actually do it from the cell that they infect. Now, this is an actual photograph of a virus called a bacteriophage. These are like, besides a tardigrade is just the coolest organism on the planet. Bacteriophages are the next favorite of mine. Not only do they look freaking cool, like an alien, they actually kind of act like an alien as well. This is a protein capsule. Inside here is the DNA or RNA, because there's different types of viruses. Some are DNA, some are RNA. And it attaches, this is a bacteria. So phage means virus, and a bacteriophage is a virus that infects bacteria. And I think I have on my Google Drive still, I screenshot a section of a Netflix series on bacteriophages. They may actually help us cure superbug infections in hospitals. And I might assign that as like a small activity on the side, but I have to make sure I find the video first. Anyway, so what happens is the bacteriophage will literally take the shaft here and stab into the membrane, and the DNA will leak into the bacterial cell. Now, once the viral DNA leaks into the bacterial cell, it will, it's basically like invasion of the body snatchers, where it takes over the whole cell activity. So the cell does nothing but then make new viruses. Pretty cool. Anyway, so what Hershey and Chase did, now that I've labeled DNA in green, I'm going to label the protein, the outer protein capsule, in red. So this is what Hershey and Chase did. Now, they knew the structure of a bacteriophage. They knew that bacteriophages infect bacteria, but they don't know whether it's protein from the virus or DNA from the virus that actually infects the bacteria. So they're going to use radioactive isotopes to label the protein and the DNA. So I've done it here in red and in green. Now, a radioactive isotope is, hopefully you remember what isotope is. It's a different version of an element on the periodic table, and radioactive means that we are able to trace it, so we can follow where it goes, either through fluorescent labeling, like it'll glow, or sometimes we need to expose it to maybe X-ray to see where it goes, something like that. So here, I actually used the right colors. All right. So the protein was labeled with radioactive sulfur. The DNA was labeled with radioactive phosphorus. And so again, with the colors, let's just say the protein was labeled red and the DNA is labeled green. Now they mixed the bacteriophage with bacteria. They allowed the viruses to infect the bacteria. And then what they do is centrifugation, which is basically putting it in a centrifuge, which is the machine that spins around really, really fast so that all of the heavy particles in the tube will sink to the bottom. Now, in the case of bacteriophages, because the bacteria invades the virus invades the bacteria with its genetic material, and we're trying to determine whether or not that's DNA or protein. Whichever one it is, whether it's the red or the green that is at the bottom of the tube with the heavy bacteria, that is going to be the genetic material.
In the Hershey-Chase experiment, bacterial viruses, called phage, were used to demonstrate that DNA is the genetic material. The phage used in this experiment consisted of a DNA molecule surrounded by a protein coat. When phage infect bacteria, they attach to the surface of the bacterium and inject the DNA into the cell. The protein coat remains on the outside of the cell. In the first part of the experiment, phage were produced in a medium containing S35 radioactively labeled amino acids.
That's a sulfur.
This resulted in a phage population with S35 labeled proteins, but no radioactive label in the DNA. The phage were then allowed to infect the bacteria. The phage attached to the bacterial cell and injected their DNA, but the radioactively labeled protein coat remained on the outside of the cell. The phage produced in these cells contained no radioactivity. Vigorous shaking caused the empty protein coats to be removed, but did not interfere with production of new phage in the cell. In the second part of the experiment, phage were produced in a medium containing P32 labeled deoxyribonucleotides. This resulted in a phage population with P32 labeled DNA, but no radioactive label in the protein. When the phage infected the bacteria, the P32 labeled DNA entered the cell and could be found in phage subsequently produced in the infected bacteria. This demonstrated that the DNA, but not the protein, carries the genetic information for a new generation of phage.
Oh, okay. So just to, what do you call that? To summarize, in the second part of their experiment, when they labeled the DNA with the phosphorus, any new viruses that are going to be made using this labeled DNA. So all they have to do is look for the new viruses that are in the bacteria, whether or not they're labeled with either sulfur or phosphorus. If it's labeled with sulfur, which is what the protein was labeled with, then we know that protein's the genetic material, but it wasn't, it was the phosphorus. So by being able to trace the genetic material from the virus to the bacteria, they were able to determine that DNA was in fact the genetic material. I hope that makes sense.